Hi, everyone. Thanks to the organisers for inviting me down to talk to you, to talk about the work that we're doing at the University of Exeter on antimicrobial resistance in the environment. I'm really glad that Matt went first because he's already introduced a lot of the concepts really well that I'm going to be talking about. So hopefully there'll be some little reminders in there. I might explain some acronyms as well, but he's mostly done all of it. I'm going to be talking about coastal waters as an example of a natural environment. Our work, I've got 3 studies here that I'd like to present, and we'll talk about the conclusions that we can draw from these studies. First of all, we know that antibiotic resistance is a real problem in human health and in veterinary medicine. A lot of the work that has been done on understanding transmission has been focused on hospital environments. This is completely understandable because there's lots of vulnerable people, there's high antimicrobial usage there, vulnerable people, vulnerable to infections. But this is just part of the picture. We've heard a bit more about antibiotic use in farm animals and their potential transmission to humans and the community. But I'm interested in the natural environment because if we can understand all the different ways that people are exposed to antibiotic-resistant bacteria, then we can perhaps come up with some sensible and effective prevention strategies to reduce transmission and prevent drug-resistant infections. Like I said, I've been studying coastal waters maybe for the past 4 years. This is because they are regularly contaminated by a variety of pollution sources. This diagram shows we've got kind of inputs from farm animals, human waste, land runoff, stormwater, all contributing billions of bacteria harboring these mobile resistance elements that Matt was talking about, plasmids with resistance genes. They also bring with them these selective compounds, so antimicrobial residues, heavy metals, and things like that might select for resistant phenotypes and genotypes in the natural environment. The second reason I'm studying coastal waters is because people go in coastal waters. They enjoy going in coastal waters for recreation, things like swimming, fishing, boating, surfing. And it's during these activities that people can come into a high degree of contact with coastal waters that are containing these potentially resistant or harmful bacteria from other humans and animals as well. So when I first started working on this topic, I think the first question I really posed was, how do we know that people get infections from the sea? And what we're really getting to at this point is, how can we demonstrate causation? What's an whether is it just association or is it actually causation? So I've got these 9 points here, and I don't really call them criteria because it makes it sound as if each and every one of them has to be demonstrated in order to prove causality, but I think they need to be considered. So if you're not familiar with these as environmental epidemiologists, these are the Bradford Hill kind of concepts for demonstrating causality, and I'll just go through them. Strength is related to the strength of the association. So if you have a large risk, if an exposure demonstrates a large risk for an outcome, it's likely to be causal. Consistency has this observation being demonstrated by different people at different times, different circumstances, different places. If it has, then it's likely to be a real observation and not just found by chance. Specificity, is this a specific location, a specific organism, a specific group of people that are affected by this particular disease? Temporality, which came first, it's just. So this is a question of, does the horse come before the cart or the cart comes before the horse? So does exposure precede the outcome observed? Biological gradient responds to a kind of dose-response relationship. So if you have a high dose of an exposure, are the risks greater or do you get a worse version of the outcome? Plausibility, it means that we have to the causal relationship has to kind of coincide with what we understand in terms of biological and the natural history of a particular disease. Kind of the same kind of thing with coherence. Experiment, so if we observe an association, between an exposure and an outcome. If we remove that exposure, do we then reduce the outcome? And the analogy, we might not see some kind of very strong evidence in a slightly similar but different field, but it might lend evidence to what we're seeing. Hope that makes sense. So taking all of these into account, I went about doing a systematic review. The systematic review, I was looking at the risk of people acquiring infections from recreational exposure to coastal waters in high-income countries. So I basically set about doing this in 2013 and update it on a regular basis. A systematic review of 40 studies, and it included a meta-analysis of the risks associated between various different outcomes. I've got gastrointestinal illness, ear ailments, symptoms of any illness, as well as infections caused by specific microorganisms. And that one's quite important because a lot of the time in these studies, they haven't isolated or understood what is the organism that's causing a particular symptom. The systematic review also allowed me to learn as well in this area, how do people measure infection? What are the indicators of infection? And I also wanted to see if anyone had been looking for antibiotic-resistant bacteria in coastal waters and indicators of infections in humans. So these are the results. They are unpublished at present, but fingers crossed soon. This is a forest plot just summarizing the meta-analysis results. So this isn't actually the meta-analysis. But what I just want to highlight very quickly is that we're seeing so for all of these, most of these outcomes, we're seeing an effect size of above 1. And for a lot of them, we're seeing really significant confidence intervals. Another outcome I'd like to draw your attention to is the one at the very bottom, ESBL producing CA, UTI, E. coli, K pneumoniae. Okay, I'll just go into a bit more detail on that in the next slide. So ESBL producing, these are antibiotic-resistant infections. Basically, they're community-acquired urinary tract infections caused by E. coli or Klebsiella pneumoniae. This study was conducted in, I think it was Norway. Yes, it was Norway. And they did a case control study of people who had community-acquired urinary tract infections caused by E. coli or Klebsiella pneumoniae. And they compared people with resistant infections to those who had susceptible infections or treatable infections. They looked at various different risk factors, and one of the risk factors they found to be associated with having a resistant infection was sea swimming. So I found this really interesting. They found that ESBL producing bacteria were associated with swimming. And ESBLs, essentially, they're extended spectrum beta-lactamases. They're an enzyme, like Matt said, a bit like a Pac-Man chomping on an antibiotic. They degrade the antibiotic, extended spectrum beta-lactam class antibiotics. And this is a large group of antibiotics, includes penicillins, third generation, fourth generation cephalosporins, as well as the carbapenems. These are kind of really important antibiotics. In fact, cephalosporins certainly are classed as essential medicines because they are the only treatment or one of a few alternatives to treat serious human infections. And as Matt did mention in his talk earlier as well, CTXMs are a particular group of these ESBLs. They're usually plasmid-borne, so they're on these genetic elements that can be spread between bacteria. And they're the most prevalent ESBL gene observed in the clinic. This is a diagram of the different ways that bacteria can share parts of their extra chromosomal DNA. The most important, I think, is probably this one at the bottom, the conjugation. I'll just do this one. This is conjugation where bacteria share a plasmid. So one bacterium will duplicate its plasmid and pass it on to another bacterium in the vicinity via this kind of conjugal pilus. And I've been looking at E. coli because these are a big health problem. they are the main cause of urinary tract infections in community-acquired urinary tract infections. They're also observed in animals as well in the environment. They're used as a fecal indicator in environment. So the Environment Agency, for example, takes water samples from bathing waters and monitors the levels of E. coli in them to make sure that there's very little fecal contamination. These E. coli are really good indicators of fecal contamination from human and animal sources because E. coli are found in the guts of both. In addition to this, E. coli readily undergo this genetic exchange of DNA. And in addition to that, obviously, some of them cause diseases. And we've heard about urinary tract infections. They also cause kind of gastrointestinal infections and meningitis and a variety of other really serious illnesses. So the second study I'm going to briefly tell you about is this study that we did to look at human exposure to and colonisation by antibiotic-resistant bacteria in coastal waters. Again, this kind of involved E. coli. Actually, it involved 3 different stages to this method. The first was an environmental assessment, where we aimed to measure the abundance of and the prevalence of E. coli that were harboring these CTXM genes. The second part was to use that information and apply it to E. coli levels found in the environment in coastal waters to estimate, on average, how many of these resistant E. coli people are swallowing when they do a particular water sport. And the third part was an epidemiological survey where we recruited an exposed group and an unexposed group to our survey and measured their fecal material for E. coli that are harboring these CTXM genes. So I'll just go into a bit of detail about each one of these steps and the results and then summarize at the end. So in the environmental assessment, the Environment Agency, which takes these samples anyway, they gave us 100 samples from a geographically kind of widespread locations around England and Wales. They gave us these water samples, and we isolated E. coli as well as resistant E. coli from these samples using culture-based methods. So this means that we just grew the bacterium out on this kind of chromogenic agar. You'll see this is the chromogenic agar, and E. coli colonies turn up blue, other Enterobacteriaceae turn up other colors like pink. So we were able to then detect the prevalence and the presence of E. coli that are resistant to this third-generation cephalosporin, cefotaxime. And then we used PCR to detect the presence of CTXM genes in our resistant E. coli isolates. And essentially, we found that resistance to cefotaxime in E. coli from coastal waters is really rare. Less than 1% of E. coli resistant to cefotaxime. And then about 2/3 of these resistant bacteria, so 0.07% of the total E. coli in coastal waters are harboring these CTXM genes. We then use this information in a formula to work out, on average, how many of these resistant bacteria are people swallowing every time they do, say, surfing or swimming. And so we use this information as P, so that's 0.07 from the previous slide. But we also got information from the Environment Agency's DataShare website on the levels of E. coli found in bathing waters around England and Wales. We then applied this to the volume of water that people generally swallow during a session of their chosen activity. And if you're interested in what exactly that means, I can answer any questions at the end of the presentation, or I've put the publication at the bottom here. So the results of this, we found that exposure is likely to be quite low, but particularly in surfers, they're at risk of swallowing about nearly 3 of these CTXM harboring E. coli every time they go surfing. So we used this information and decided to recruit 150 regular surfers. By regular, I mean they surf about 3 or more times per month even. And we wanted to recruit 150 matched controls, and they were matched by age, sex, geographical location. And then we measured their gut bacteria. We looked at their gut bacteria to see if they were harboring resistant E. coli. So the data collection of this, which we lovingly called the Beach Bum Survey, involved people responding to a questionnaire which collected information about various risk factors associated with gut colonization by resistant bacteria. So, for example, any recent travel, their diet, animal ownership, things like that. And we asked them to collect their own fecal swab, which they then sent back to us in post. No one was bending over. Not with me, though, anyway. With the fecal swab, we then plated the bacteria from the swabs out onto the same chromogenic agar, supplemented with cefotaxime. And basically, we marked surfers or non-surfers as having resistant bacteria present or absent. We used the same method as before to identify colonies that were harboring the CTXM gene using this PCR method. So this slide I've put in because it shows kind of how, first of all, it tells you how interested people were in taking part in this survey. I mean, it was difficult to get people to part with their poo, but we did have a lot of interest in this study. And I think that was partly due to our kind of working with Surfers Against Sewage. That's SAS at the top. So our recruitment strategy was to work with Surfers Against Sewage, and they would tell their members, their surfer members, about the existence of the study. People who would be interested would come forward to volunteer, and then a member of staff at Surfers Against Sewage screened interested participants for their eligibility. And we had a number of eligibility criteria because we wanted to be more sure that, couldn't be certain, but we wanted to kind of eliminate some other sources of reasons that people could be carrying resistant E. coli. And these were things like they can't have travelled outside of the UK in the past 6 months, no overnight stay at a hospital in the past 6 months, no recent antibiotic use in the past 6 months, and they had to be over the age of 18 and things like that. So 308 people were deemed eligible, and so about double that was sent out because we wanted surfers to recruit their own match control so they could find a friend who wasn't a surfer, who was the same age, same sex, lived in the same area, to do the collection for us and then send everything back together. So we got nearly 300 swabs and questionnaires, a fairly even split between surfers and controls. But we had to let some of them go because they did actually write on their paperwork that they weren't actually eligible. So we had 273 swabs analysed, 143 from surfers, 130 from controls. And these are our results, again, unpublished, but it shows that surfers are maybe nearly 3 times as likely as non-surfers to be harbouring E. coli that are phenotypically resistant to cefotaxime. So this means they're able to grow in the presence of the clinical break point of that antibiotic. And then the bottom row shows that they're about 4 times as likely as non-surfers to be harbouring E. coli that are producing the CTXM, this enzyme.